So today we're talking about blood. Our learning objectives for today, we're going to go through the functions and composition of blood itself, the physical characteristics of it. We'll define what a hematocrit is. That should be kind of review for most of you who are already in lab. We'll go through each of the formed elements, talk about what they do, what their relative abundance is, et cetera, et cetera. And then we'll go through blood plasma, what it is, its function, and then we'll explain how the formed elements are formed and where they come from, what their lineages are, why are they different, et cetera, right? Because we talk about red blood cells, white blood cells, platelets. Well, how are they all related? They are kind of all related. They're all formed in the bone marrow, but they come from different lineages, as we call them, okay? So blood itself is a continuously regenerated connective tissue. From last semester, you went over connective tissue. What were the characteristics of connective tissue? What are some of the characteristics that define connective tissue? From last semester, there were actually three things. Connective tissue consists of cells separated by an extracellular matrix. Okay, that extracellular matrix is what we call ground substance. Okay, so like, give me an example of a connective tissue that you know of, besides blood, Christian. Huh?
Loose connective tissue.
All right, loose connective tissue. What else? What does the loose connective tissue describe in that case? The loose part of the connective tissue describes what? As opposed to like dense connective tissue. Close. Not the amount of layers, and not necessarily layers, but close with the amount, but layers of what though? All connective tissue had collagen, elastic, or reticular fibers, right? So all connective tissue had fibers. So loose, instead of layers of fibers, okay? So loose means they're kind of spread out. So all connective tissue has cells separated by a ground substance, and then they has to have some type of fibers, right? So blood being a liquid connective tissue, what is the extracellular matrix that is blood? Is it a solid? Is it a liquid? Is it a semi-solid? Well, it's not a solid, right? Because blood flows. It's a liquid. Plasma is the liquid that the blood cells float in. That is the extracellular matrix of the connective tissue of blood. Does that make sense? Yes? So my question then is, if blood is a liquid connective tissue that continuously flows, it is a connective tissue, so it has fibers. Where will you find those fibers? Think about what blood does. What does it do? Well, it moves gases, nutrients, waste, hormones. So obviously it's big in transport, right, through the cardiovascular system. Great. What else does blood do? It clots. Inevitably, you will experience a broken blood vessel. Yes? So, biologically have evolved and created the system that shuts off the opening when the broken vessel to prevent the loss of blood. So clotting is a property of the blood tissue itself, and that is where the fibers come in. Does that make sense? So parts of the blood, proteins and cells, fragments that are present in the blood itself become activated when a blood vessel is ruptured or broken. That activation leads to the conversion of a soluble form of fiber to an insoluble form, leading to a clot. Right? That's where the connective tissue comes in, or the fibers of the connective tissue comes in. Right? And we'll talk more about clotting, hemostasis later in this chapter. All right, so transport, clotting, right? Transport is important, right? Blood moves through the cardiovascular system, through vessels, or a series of arteries, capillaries, and veins. Arteries away. Arteries typically carry oxygenated blood away from the heart, A for away. Veins typically carry deoxygenated blood back towards the heart, okay? And capillaries is where there's an exchange. Does that make sense? Yes? Okay, so we'll go over that blood vessel, the movement of blood through blood vessels when we talk about vessels themselves. All right, so but for right now, arteries, veins, and capillaries. Right, and capillaries are where the gas exchange occurs. Now, the components of blood, which I've already kind of alluded to, two parts. The formed elements, which are your cells, red blood cells, white blood cells, and then cell fragments or platelets compose your formed elements. All right, so erythrocytes are red blood cells. Their job is to transport respiratory gases in blood. More specifically, or most specifically, they are responsible for the transport of oxygen. Okay, that's the big one. Do red blood cells also carry a little bit of carbon dioxide? Yes, they do. All right, but their main function, like 99.99% of the gases that they are transporting is oxygen. Okay, that's its main job. Carry oxygen, deliver it to tissues, right, and go from there. Okay? Leukocytes are all of your white blood cells. These include neutrophils, lymphocytes, monocytes, basophils, eosinophils, macrophages. Those are all white blood cells. Their job is they compose the workers, they are the workers of your immune system. So they work and they defend against pathogens, both non-specific and specific immunity. What that means is I pick up a Staph aureus infection or a bacteria from the skin into one of my little cuts on my hand, right? There are macrophages and neutrophils that are present right there. They recognize that. They get rid of it right away. That is a non-specific, right? Doesn't matter which bacteria comes in. They just recognize it, engulf it, they'll break it down, okay? A more specific type of immunity, I'm exposed to a flu virus. Over time, I might develop antibodies to that flu virus. The lymphocytes recognize that flu virus, all right, respond to it, immune response. That is a more specific immunity. We'll go through immune system in a few weeks, all right? But together, leukocytes help to prevent and work against pathogens, right? And then platelets, which are also called thrombocytes. I'm gonna put these in purple. Right, platelets are also called thrombocytes. They help to form clots, right, that prevent blood loss. And the clot is also called a thrombus. All right, so erythrocytes and leukocytes are full cells. Thrombocytes, kind of a misnomer. Cyte really means cell, right? But thrombo refers to clot. Platelets are actually cell fragments, right? So pieces of cells that are just present that the precursor cell has kind of like blown up and released these little pieces, okay? So those consist of your formed elements, and plasma is the fluid portion of blood, which contains plasma proteins and dissolved solutes, and is mostly about 90% water. Okay? So on the right here, this is a centrifuge piece of sample of blood. This, after it's been spun down, is the plasma. in red are that is called your hematocrit. Okay? The hematocrit is equal to the amount of red blood cells in a volume of blood. All right? In some cases, you might see in older literature, you might see something called the packed cell volume PCV. That is also the same number of red blood cells in a volume of blood. Okay? And then this little piece right here, that is what we call the buffy coat. That is where all of the white blood cells are found, in that buffy coat. Right? The plasma represents about 55% of a normal blood volume. The hematocrit is about 44%, and the buffy coat is about 1% of the normal blood volume. If we take a drop of blood and we look at it under the microscope, we can observe the different formed elements and what they look like, right? First off, you will see lots of red blood cells. All of these are red blood cells, right? All of these things, right? I'm not going to circle them all, but they kind of look like donuts, right? They have a ring and they look like a space in the middle. That's because the red blood cells are shaped as what we call biconcave discs. On either side, they are pushed in. All right, and that structure is important for its function. The biconcave disc leads to an increase in the ratio between surface area and volume of the red blood cell. It allows the red blood cell to be flexible as they move through these capillaries, right? The surface area to volume ratio, that proportion is important in how fast oxygen and carbon dioxide diffuse across the membrane, okay? By increasing that ratio, it allows oxygen to be dropped off to tissues really quickly, okay? And that's really important because blood doesn't stop flowing, right? So as red blood cells move through capillaries, they slow down, but they don't stop. They slow down, exchange occurs, and they move back. Does that make sense? So if that ratio of surface area to volume was off, the speed of transfer would be less, and then it would cause problems. Does that make sense? Whenever blood slows down, transfer doesn't happen. The tissues don't get the oxygen they need, right? So you know how this feels like. You sit in the chair, legs crossed. After a little while, maybe your foot starts tingling. Yes? Well, what's that from? You cut off and you slow down blood going through the leg, through one of the vessels. Blood kind of gets trapped in the tissue, in the extremity. The tingling is a message to your body saying, we need to clear things out. Oxygen has to get here. Got to get rid of carbon dioxide because blood flow is not happening. Okay? So we want to optimize blood flow through a tissue to allow for exchange to occur. And red blood cells are structured in that way to have that happen very quickly and efficiently. Okay? Then we have these are erythrocytes, all of those, right? Red blood cells, those are your biconcave discs. We have a couple neutrophils. These are the larger white blood cells. We'll talk about their structure in a little bit. Neutrophils are the most abundant white blood cell, right? In a normal, quote unquote, normal blood sample, right? So someone who's not experienced, doesn't have an infection, not sick, right? Normal, healthy individual would have mostly neutrophils. Okay, if someone had less neutrophils than normal, a normal percentage, and maybe had more eosinophils, that's pretty much indicative of something is off. Does that make sense? Right. So we use these normal ratios, normal percentages in a way of diagnosing possibilities of things going on in the body. All right, and then finally we have platelets. Platelets are these pieces right here, okay? And this little thing, if you can see that, okay? See that kind of blob? It looks like it's not really a cell, but it kind of looks like a cell, right? That is the cell that actually is breaking up and separating into pieces, turning into the platelets. That's why we have so many platelets here, because this cell, what was the cell, is starting to break down and producing the cell fragments that are platelets. Okay? So erythrocytes, by far the most abundant cell in a blood sample. Neutrophils, most abundant white blood cell. And then platelets are cell fragments. Questions on blood smear, what you might see on that. So, obviously, we've talked about two distinct functions for blood. Transportation. It transports gases. It transports wastes. It transports nutrients like glucose, right? It transports hormones. On top of that, it also transports heat, right? So we go outside and we're having a lovely snowball fight that all of the students had on Monday, yeah, right? Or you go sledding and fall down, right? What happens? You're out in the cold. What happens to your extremities, your fingers? What? They become red, right? They get cold first, and they're probably going to be a little bit pale, right? That's because the blood vessels constrict, and we transport, we hold in heat other places. But then you come back inside, and what happens, Jackie? Your fingers get red, right? That redness is those blood vessels open up, more blood flows through, returning heat to those tissues to help warm them up, right? And so that's important as well. Okay? The other function that we've kind of alluded to is protection. Leukocytes protect against pathogens and disease, right? Plasma proteins are also part of this. Within the plasma, you have these non-specific proteins like lysozyme and complement that work to identify potential pathogens and get rid of them right away before your immune system even recognizes they're there, okay? And then platelets and certain other plasma proteins like fibrinogen and fibrin and thrombin, right? Those help protect against blood loss through the clotting process. The clotting process is called hemostasis. And there's 4 steps to hemostasis that we will discuss later. Blood also helps regulate things, all right? Regulates body temperature. Okay, so what happens when you're sick and you get an elevated body temperature? You have a what? Fever. What happens to your face when you have a fever? It's hot. Why is it hot to the touch? Well, blood vessels open and blood is at the surface. Why is the blood at the surface and why are you warm to the touch? What's the point of that? It's the same thing that happens when you run around and you're exercising and you sweat. Well, the point of that is to help reduce and dissipate heat to the environment to help lower your body temperature, right? So your body, when you have a fever, your body recognizes this extra warm. It's warm, right? Body temperature's elevated. Your face gets flushed. You might have like kind of cold sweats. Okay, that's because your body recognizes my body temperature is high. I need to open the blood vessels and remove heat from the internal out to the outside. Just like you are when you're running around sweating, right? Flush, sweat, move, dissipate heat. That is regulation of body temperature, and the blood is part of that, okay? Blood also helps regulate your body pH. So pH of the blood is about 7.35 to 7.45. Okay? That's the normal pH of blood. Okay? That happens to be the normal pH of almost everything in the body, right? Outside of like things like the stomach and some other places. That pH is where the enzymes of your body at the right temperature work at their optimal rate. They break things down, they build things up, right? All the reactions that happen in your body kind of have been optimized to happen within that range in temperature. Okay? So if things go up above that, your blood works to bring it back down. If it goes below 7.35, your blood, in conjunction with other systems, work to bring it back up. Okay? So your blood has this innate buffer system and prevents big swings or changes in body pH, right? By sequestering hydrogen ions and releasing bicarbonate ions, etc. Okay? And also with that is somewhat related is fluid balance. Okay? Your blood, there's a closed system. Your blood vessels should be closed, right? You have a certain volume of blood that you're walking around with all the time. Yes? And you will lose some water to the environment, and some water may come out of the blood through plasma loss, right? But water is added to the blood through absorption in the GI tract, right? And lost through urine, skin, sweat, right? And respiration, breathing. Okay? So the fluid exchange occurs between the blood and the interstitial fluid. Interstitial fluid is the fluid around cells, not fluid around cells. Okay? That fluid is important in transfer of materials. Right? It allows the cells to allow for diffusion to occur. Okay? And so the blood helps maintain that osmotic balance. Right? No, you never want to have like too much stuff on one side or the other, because then fluid doesn't go the right way. Right? What happens when fluid accumulates in a tissue? What is that called? Anyone know? When fluid accumulates in the tissue, we say it what? Not. So let's say if I notice it's swelling, right? You hit something, it swells, right, or whatever. So swelling is just an accumulation of fluid in the tissue. Okay. Well, how did that happen? It could be if there's not like a traumatic injury or something, right? It could be that there's a problem with fluid exchange, and fluid is only getting trapped on one side or the other. Okay? We want it to be pretty much equal. We want, plenty of hydration come in, delivering fluid to the tissues, and then whatever's left over gets picked up and returned back to the blood, right, as through the lymphatic system. That's the ideal. Taylor.
I just have a question about the body.
Okay.
You said that when the blood absorbs the heat from the body, it's vasoconstriction, and then when it releases heat from the body, it's vasodilation.
So, not necessarily. So, when the blood vessels dilate, they open, more blood flows through the vessels. So, that allows, especially at the surface of the skin, the capillaries dilate, more blood is at the surface, that will dissipate through the heat to the environment. When you're cold, the vessels constrict, less blood at the surface, more blood internally. You conserve body heat. Okay? No, stop. All right, so some physical characteristics of blood. We already mentioned one. pH of blood is about 7.35 to 7.45. Blood is a lovely red color, right? Oxygen-rich blood is bright red, like that sweatshirt. She's on her phone. That sweatshirt, bright red, scarlet red, Ohio State red, okay? I hate Ohio State, sorry. Okay? That is oxygen rich. Oxygen poor blood is a little darker, more like a crimson, like Madeline's sweatshirt, maybe even a little darker. Okay? Think Alabama Crimson Tide colors. Okay? I don't like them either. Okay? And that is a result of the hemoglobin that is present in blood, in red blood cells. When oxygen is bound to hemoglobin, for whatever reason, it shines this scarlet red color. Okay, and you can see the difference here in these two vials. All right, so scarlet red, oxygen rich, dark red, oxygen poor. You've all had a blood sample taken at some point in time, yes? Okay, is it oxygen rich blood or oxygen poor blood that they're taking out? Think about it. You've looked at that vial, if you can stomach it, right, and you see what it looks like. Is it oxygen rich or oxygen poor? Oxygen poor. It's coming from a vein. Okay? So most veins carry oxygen poor blood, so it's going to look like that dark red color. Okay? If you were to puncture an artery, it'd be too high pressure. That's why we don't do it. It would be bright red. Okay? Volume of blood from females about 4 to 5 liters, males about 5 to 6. Why is it a difference? Mainly in size. Right? So different, more muscle mass, usually more body mass as a whole. Right? So we have higher blood volume. Viscosity. What is viscosity? It's thickness, right? It's the difference between water and maple syrup. Right? We know how thick and how much maple syrup kind of doesn't move, right? Why does maple syrup not move that much? Well, it has a lot of sugar in it and other stuff, right? More stuff you have in it, the thicker it is. Does that make sense? Now, blood has stuff in it. It's plasma. It's got proteins dissolved in it. It's got red blood cells. It's got white blood cells, etc. So blood isn't like water that is just water. Okay, we use water as a relative standard base point, starting point, comparison. So in comparison to water, blood has more stuff, so it's got to be more viscous. That's why the 4.5 to 5.5 is the measurement. Okay? Plasma concentration about 0.9%, 0.09%. Temperature is about 38 degrees Celsius, and pH about 7.35 to 7.45. Why is the temperature of blood 38 degrees Celsius, but your body temperature is 98.6 or set 37 degrees Celsius? Blood's a little warmer because it has to transfer heat to the tissues. Right, so it should be a little bit warmer. So volume, viscosity, right? So viscosity, blood is about 4.5 to 5.5 number was, about 4 to 5 times thicker than water. And we again, based upon how much stuff is there, all right? So how much stuff is dissolved in the fluid that we're talking about makes it more viscous. The more red blood cells you have, viscosity goes up. The more white blood cells you have, less of an importance, but the viscosity could go up, right? If the amount of fluid decreases, viscosity increases. How would the amount of fluid decrease? What condition you might happen in the body that might lead to a decrease in fluid? Taylor. Dehydration. Remember, plasma is how much? What percentage water? About 90% water. So if your body's dehydrated, you could lose fluid from your tissues. You could lose fluid from the blood. It's not going to come from the red blood cells. It's going to come from the plasma. Less plasma, right? Less volume that the foreign elements are going to float in. That means there's a higher percentage of red blood cells. that makes it more viscous. Okay? Other examples of like increasing viscosity, if someone is taking hormone replacement, testosterone. Testosterone leads to the formation of more red blood cells. If they're taking a hormone such as erythropoietin, or if they have kidney issues, you might produce more erythropoietin, which stimulates the production of red blood cells. More red blood cells, higher viscosity. Temperature, body pH, etc. Questions? All right, here's centrifuge blood. All right, put it in a centrifuge, plasma, 55, we already went all of this. Questions? Molly, you're my buffer. You're my measuring stick, Molly. See if everyone's paying attention. Any questions? So you may have seen this. All right, so again, here you have your 92% by weight water from plasma. Right, plasma represents 55% of whole blood. And then in this Buffy coat, in this picture I like, all right, that 1% of those leukocytes, it breaks it down to where you will find the platelets are part of that 1%, that Buffy coat, and the white blood cells are part of that 1% or that Buffy coat. And within that 1%, you have 5 different types of white blood cells that you will find in a normal blood sample. Neutrophils, lymphocytes, monocytes, eosinophils, and basophils, in that order of relative abundance. Okay? So the neutrophils are going to be the most abundant white blood cell you find in a normal blood sample. Okay? And then by far the most amount of blood is going to be, or most amount of formed elements is going to be red blood cells. Questions? So we talked about the red blood cell here, that packed cell volume being your hematocrit. You did this in lab this week or last week, yes? All right, so what did you find out about the blood sample you used? What was your, if you remember, what was your hematocrit percentage? Was it normal at 44%? Taylor?
No.
Why not?
Well, ours we did today, and it's because the blood was like
Blood's a week old, that's number one. What else? Well, it's also bovine blood, so it's not human blood, right? So that might be a little bit different, but the week old plays a big role in it, okay? So your hematocrit is percentage of all the formed elements. Technically, clinically, it's the percentage of only the red blood cells, right? And so you did this in lab, you measured the full length, the full volume of the total blood sample, right? Use that measurement in order to measure the red blood cells, divide the red blood cell measurement by the total measurement, you get a fraction, and that gives you your percentage of total volume, okay? Now, adult males have about a 42 to 56% normal hematocrit, and females about 38 to 46. Part of the reason for that is testosterone is a very big driver of the formation of red blood cells. Okay, so testosterone is important for inducing muscle mass, right? And then we have to kind of go from there, more muscle mass, higher, more body mass index, more blood volume, and then also increase the number of red blood cells. All right, so testosterone causes more erythropoietin secretion by the kidney, right? That also is problematic because men are also disproportionately affected by cardiac disease, right? Cardiovascular disease, yes? High blood pressure, heart disease, diabetes. What happens when testosterone goes up, hematocrit goes up, and you have cardiac disease? What happens? More blood. It could lead to a heart attack, right? More red blood cells, a higher percentage makes the blood more viscous, okay? then that then puts pressure on the heart to continually move blood through the system. The more viscous, the more force has to be generated, and that can put stress on the muscles from there. Right? It could also lead to clotting issues, etc. All right, so this was a blood smear. You did this, you only looked at the pictures, but you did this in lab, right? And this allows you to visualize the different cells that are present in the blood sample. So plasma, 92% water, 7% plasma proteins. It is an extracellular fluid, okay? And it has a similar composition to the fluid that's found around cells, but it has a higher protein concentration. And this higher protein concentration is mainly due to the presence of what we call albumin. Okay? The reason and the importance for having the higher protein concentration is that it maintains the osmotic balance. Okay? So more proteins means more solutes that are present in the blood as opposed to the interstitial fluid. Think about the interstitial fluid. It's got cells, it's got some other stuff, the stuff's being released by cells, etc. So if there was not the presence of these proteins, osmotically, water would always want to go from higher percentage of water to lower percentage of water. So it would always want to move out of the blood into the tissues. We don't want that to happen, right? We have to maintain blood volume in order to maintain blood pressure and deliver all of the blood and transport all the things we've been talking about, okay? We still need to deliver fluid to tissues, but it has to be at the blood's designation, right? The blood has to decide how much fluid goes because they have other higher priorities, a la, heart beating, it's life, right? So the albumins are present to help maintain that osmotic balance and not to lose water or fluid to the tissues consistently. Right? Think about what would happen if that didn't happen, right? Blood would move through the system, it would lose fluid to the tissues, and then you wouldn't get enough back to the heart, and that's not coincident with life. All right, so by that premise, blood is what we call a colloid. It is a solution of dispersed proteins, right? Albumin is the big one that helps maintain osmotic balance. Globulins, such as antibodies that are produced by lymphocytes and plasma cells, those are present in soluble form in the plasma. Fibrinogen is an inactive form of fibrin, which is a clotting protein, right? That's inactive. We want it to be inactive so that when we're not producing clots without a broken vessel, right? That's a clotting abnormality that can lead to pulmonary embolisms, etc. Okay? There's some enzymes and some hormones that are also present in the plasma. Most of these are produced by the liver, right? And others are produced by leukocytes or other organs such as hormones. But these first, albumin and fibrinogen, right, and some of the globulins, they're produced by the liver, like leukocytes produce the globulins. But albumin is a big one for the liver and liver function. Okay? Questions? So these proteins, like I said, exert what we call colloid osmotic pressure. All right, so the colloid, the dispersion of proteins, osmotic pressure, right? So that dispersion of proteins helps regulate the movement of fluid or water across the membrane, or across between the blood and the tissues. And that helps prevent the loss of fluid from the blood as it moves through the capillaries, right? And so the job of that is to maintain blood volume and blood pressure, okay? Because blood volume is directly related to how much water you drink, how hydrated you are, right? You get water in equals water out, okay? You drink more water, you consume more water, you fill up what you need in the body, and then excess is removed through the kidneys. Does that make sense? Right? You guys know kind of You drink 8 glasses or more of water, you end up producing more urine, right? Okay. That water in, water out means you're fulfilling all the needs that are represented by the body, okay? If we lost it to the tissues, we would have to consistently consume fluid, okay? And this could be increased and altered in different diseases, resulting in fluid loss, right, from the blood and tissue swelling, right? Liver diseases that decrease the production of plasma proteins, like cirrhosis, hepatitis, can alter the formation of the albumins. If you don't produce enough albumins, guess what? You lose a lot of water and can lead to problems in blood pressure, etc. Okay? And then other kidney diseases that increase the elimination of plasma proteins. Diabetes is one, right? That can lead, that's not necessarily a kidney disease, but higher blood sugar, the kidneys start to secrete and excrete glucose from the blood. So they work overtime. With that also goes fluid from the blood and some of the plasma proteins that go with it. So your albumins are the smallest and the most abundant group of proteins. And like I said, they exert the greatest colloid osmotic pressure, right? Their job is they're actually transport proteins and allow lipids, hormones, and ions to be transported in the blood, okay? Remember, blood is an aqueous solution. It's mostly water. So some lipids and other lipid-soluble hormones, they don't travel well in the blood because of the water, the hydrophobicity of the water, of the lipid itself. Okay, so it has to be connected to and transported by a protein, an albumin. Globulins are your second largest group. They're smaller alpha and beta ones, right? They also are involved in transport. And then your gamma globulins are your antibodies. All right, and that's part of the body's defenses. So the function of the albumins and the globulins, transport stuff, right, that needs to be transported through there, but also help regulate colloid osmotic pressure. Fibrinogen makes up only about 4% of plasma proteins, and its job, it contributes to clot formation, right? It's transferred. So fibrinogen, see this ogen, that end. Every time you see that, you basically know that that's an inactive form of a protein. Okay, we might talk about fibrinogen, we'll talk about chymotrypsin, we'll talk about trypsinogen, right? Those are inactive forms of a protein. So when you take that off, another enzyme is going to cleave and convert the inactive form to the active form. So fibrin is the active form, that's your fiber, right? And that is a insoluble fibrin strands. That's the fibers that are related to a clot. And then finally, we have less than 1% of proteins are your regulatory proteins, enzymes and hormones that are important for the body. Okay, so by far, like, importance in terms of proteins, albumins, globulins, and then fibrinogen for hemostasis and clot. Other stuff that is present in the blood, right? So we talked about blood being a colloid. It's a suspension of stuff, but it's also a solution. There are things that are dissolved in the plasma, right? Organic and inorganic solutes, molecules. Things like electrolytes, chlorine ions, calcium ions, sodium ions, nutrients such as glucose, gases, carbon dioxide and oxygen, right? And waste products, right, are such as carbon dioxide. Polar or charged substances dissolve easily. Why would a polar or a charged substance dissolve easily? Why would a polar or charged substance dissolve easily? Why would a polar or a charged substance dissolve easily? Okay, how does that work in terms of blood though? Why would it dissolve easily? It's polar, so why does it dissolve easily in the blood though? Because blood is a aqueous solution. Aqueous being water, which is also a polar molecule, right? So, is that what you were getting at?
Yeah.
Oh, yeah, good, Christian. Yeah, good. And you said charged, that's what, yeah, okay, good. Right? Because of the aqueous nature of blood as a solution, that is why polar or charged substances dissolve easily, and that's why non-polar molecules and lipids require the carrier proteins. Questions? Questions? So are we good on hematocrit? Role of proteins in plasma? Role of the formed elements? Yes. Yes? Okay. So where do the formed elements, where are they produced? You can remember back to last semester. Where are the formed elements or the produced? In the bone marrow. What type of bone marrow? Red bone marrow. Okay? So the production of blood cells is a process we call hematopoiesis or hemopoiesis. They're both the same thing. You might see them used interchangeably depending upon what literature you're reading. Right? So hematopoiesis or hemopoiesis, same thing. It is the production of the formed elements. And then we can break it down into different parts. We could call about leukopoiesis, production of white blood cells. Erythropoiesis, production of red blood cells.
What are you going to use?
I will use both of them in different places. So like you might see hemopoiesis in your Connect. You will likely see hematopoiesis on anything that I write for like an exam or something. Okay? So it occurs in the red bone marrow of certain bones, mostly flat bones, okay? And what it starts with, this process starts with a stem cell. That stem cell is called a hematocytoblast, okay? That can and does give rise to all of the formed elements, red blood cells and white blood cells. So that is a common ancestor of all of this blood, of all of the formed elements. Okay? This is a pluripotent stem cell, so it can differentiate into many cell types, right? But it divides into two separate lines, okay? The myeloid line and the lymphoid line. Lymphoid gives rise to, undo that, right, is the ancestor for your lymphocytes. Okay, mostly your lymphocytes, your myeloid line, mostly your red blood cells, etc. So there's two separate lines, right? So we have a common stem cell, and then it starts to differentiate as we move down that line. The myeloid line, erythrocytes, all the leukocytes except for lymphocytes, and the megakaryotes. The megakaryocytes, those are the cells that produce platelets. Does that make sense? The lymphoid line only produces lymphocytes. Okay? So on this slide, you see a bunch of different growth factors. I don't want you to concern yourself with knowing the growth factors. All right, I'm not going to ask you to identify what does colony stimulating factor 1 do? What does macrophage colony stimulating factor do? Right? You can have an idea of what they do. But the point being is there are a number of steps that occur between your first megakaryocyte, or your megakaryoblast, right? And then the myeloid and lymphoid lines, there are multiple steps. Each one of those steps in that lineage, eventually getting to a mature red blood cell or a mature white blood neutrophil, is regulated by the presence of these growth factors. All right, so if you have a macrophage colony stimulating factor, that stimulates the precursor cell to differentiate into a macrophage. Clear? But again, I'm not going to ask you identify what a granulocyte colony stimulating factor does. Right? That's not the way I do my exams. But the point being is this is kind of what I want you to understand that. There is one on here that you need to know, and that's this one right here. Erythropoietin is a hormone. It's also called EPO. It is a hormone that is produced and released by the kidneys. Okay? Erythropoietin stimulates the release and the production of red blood cells. More erythropoietin, more red blood cells. So your kidneys help to always, the kidneys are always kind of taking a measurement of how much oxygen is present in the blood. Okay? If there's enough, they don't release any more erythropoietin. But if the amount of oxygen that's present or hemoglobin that's bound to oxygen is present, it starts to go lower than normal, the kidneys will, in response, release erythropoietin. That stimulates the bone marrow to produce more red blood cells. More red blood cells, more oxygen bound to hemoglobin, right? And so it's a negative feedback loop. So oxygen bound will go up, that will cut off the production of erythropoietin. Does that make sense? Yeah? So erythropoiesis requires things like iron, B vitamins, amino acids. Why is iron important? Why is B vitamins important? And what happens when you don't have enough of those? Why is iron important, number one? For the production of hemoglobin. Iron is a necessary molecule for hemoglobin in order to bind oxygen. Okay? So if I don't have enough iron in my diet, what happens to the amount of hemoglobin I can produce? Not enough iron, not enough hemoglobin. Not enough hemoglobin, what happens to the number of red blood cells I have? Or what am I experiencing in that sense? Those of you who are in my lab today should know. What is that called? Anemia. Anemia is a general term for a reduced ability of the blood to bind and transport oxygen. Okay? So we could have, the one I just described was deficiency in iron. That's iron deficiency anemia. Make sense? Yes? We could also have anemia that's related to a deficiency in B complex vitamins, some amino acids, genetic traits such as sickle cell anemia, large losses of blood, right, are examples of anemia. Okay? But to overcome that, your body will recognize that, release erythropoietin, and stimulate erythropoiesis. That will begin with the myeloid stem cell, go down the myeloid stem thing, go through a pro-erythroblast, eventually becomes what's called a reticulocyte. A reticulocyte is an immature red blood cell. Okay, it lacks the organelles, it just cannot produce enough hemoglobin yet. And then eventually the mature erythrocyte is formed. Okay? Questions? Now, leukopoietin, you'll see this, the table in a little bit that actually breaks everything down, right? Leukopoiesis is the production of leukocytes, right? And it involves the maturation of a couple different types of cells, granulocytes and agranulocytes, monocytes, and lymphocytes, okay? Your granulocytes are the neutrophils, basophils, and eosinophils you will see in a normal blood cell, right? And they derive from the myeloid stem cell line. Monocytes are also derived from the myeloid stem cell line, right? They need macrophage colony stimulating factor, right? And it forms a monocyte, a large cell. And then your lymphocytes are from the lymphoid stem cell, so a different line, and they differentiate into B lymphoblasts and T lymphoblasts, otherwise known as B cells and T cells. Why are they named B and T? Reference where they mature. B cells mature in the bone marrow. T cells mature in the thymus. Okay, and some lymphoid cells actually differentiate directly into a different type of cell called a different type of immune cell called a natural killer cell. And we'll talk about those when we go through the immune system in more detail and what they do. Okay, but here's the overall idea. Okay, everything you have, this is the separation. Right, that red line is lymphoid versus myeloid stem cell. Everything on the left of that red line is the myeloid stem cell line, right? The myeloid line comes from a myeloid stem cell. They're related. They break down depending upon what growth factors are present will stimulate the cell to go which way, and the presence of those growth factors depends upon what's needed by the body, right? So eventually you get through erythropoietins up here, right? You have more of it there. Stimulates the production of red blood cells, right? Thrombopoiesis is also through the myeloid stem cell line. What's thrombopoiesis? It's the production of platelets. Okay, so erythropoiesis, the production of red blood cells, leukopoiesis, the production of white blood cells, and thrombopoiesis, the production of platelets. Eventually you get down to this megakaryocyte, which then starts to break up and perform platelets. And then in the middle here, you have production of white blood cells, such as the eosinophils, basophils, and neutrophils, and the monocyte. And all of the lymphocytes are derived from the lymphoid stem cell line. Okay?
The white blood cells was what?
You have these white blood cells, eosinophils, basophils, neutrophils, and monocytes are myeloid. All of the lymphocytes are from the lymphoid stem cell line. Okay? So are you familiar with any diseases of the blood? Sickle cell is one. Sickle cell has to do with the production of the form of red blood cells, and we'll talk about that on Wednesday. Other blood-related diseases, are you familiar with? Anyone? Have you? Hemophilia, we'll talk about that on Wednesday as well, right? The problem with hemostasis and the inability to clot. One that's related to this is leukemia. All right? So there are multiple forms of leukemia, and they are pretty much called like AML, ALL, CLL, and CML. That stands for chronic myeloid leukemia. So what line are you expecting to see in a myeloid leukemia? Well, it's going to be on the myeloid stem cell line, right? Leukemia can be either an overproduction of cells that are basically one of the cells in this lineage are growing too fast, and that leads to immature, ineffective, dysfunctional cells. Particularly, let's say a CML would be like a red blood cells. This red blood cells don't work very well, right? If you have red blood cells that don't work very well, That one of the conditions might be anemia, right? Lots of red blood cells, but when you have to look at it under the on blood smear under the microscope. Another line would be a CLL or an ALL, acute or chronic lymphoid leukemia. That's through the production of the lymphoid line. Does that make sense? Right, so you may see when you talk about, when you read and see leukemia, you may see like a chronic versus acute, lymphoid versus myeloid. You know what you're talking, you understand kind of what they're getting at. Okay? And there's your thrombopoiesis, your megakaryoblast into a megakaryocyte. And its production breaks down into the formation of platelets. Questions for today?